Hi, guys. Hope you all had a good break. We're going to be moving into our next unit, which is going to cover protein transport and cell signaling. So, we're kind of moving into the part of the semester where you probably have not studied these concepts before. The beginning of the semester, really the first half of the semester, is kind of adding on to concepts that you have already learned: structures of cells, photosynthesis, cellular respiration. All of those topics were covered in previous classes. We're now going to be moving into things that are a little probably more new to you. So do be aware of that if you have felt in the class that, oh, I know what this is, I know what this is, I know what this is, that you may go into this part and begin to feel like, oh, I don't know what this is. And you may need to spend a little bit more time with it. So, we're going to start with the concept of protein transport in cells. So, we've talked about the cell. We know we've got all of these organelles inside of the cell, and we have all of these compartments that we need to get proteins in and out of. So, when we talk about what's called the endomembrane system, so this portion right here, we're talking about basically the nucleus, the endoplasmic reticulum, the Golgi, the different vesicles that are in here that all need to exchange proteins. And we're going to have to look at how we get these proteins from one place to the other in the cell. We're also going to take a look at protein transport in and out of the mitochondria and chloroplasts as well. While they are not part of the endomembrane system, they definitely need proteins coming into them. So we are going to take a look at those too. So this is just kind of a reminder of the organelles and what they're doing. So obviously we know that the nucleus is where we've got our genetic material located, and it's where we go through the replication of DNA and transcription, things of that nature. We know the ER is going to deal with protein synthesis, lipid synthesis, things like that. The Golgi is going to direct traffic in the cell. So we've already talked about most of these. The mitochondria and chloroplasts are on here. However, again, they are not actually part of the endomembrane system, but they are membrane enclosed organelles. So let's take a look at how that happened. And we've talked about the process of endosymbiosis before. We have talked about how we had a eukaryotic cell, and at some point there was a prokaryotic cell that had the ability to utilize oxygen. So we had an aerobic prokaryotic cell that was essentially engulfed by this eukaryotic cell. And because of that, the plasma membrane came around and essentially surrounded this entire thing. So if we think about this prokaryotic cell had its own cell membrane, and then it ended up being wrapped in another cell membrane as it was engulfed by this cell. So when we talk about the mitochondria and the chloroplasts, these are the organelles that were engulfed and are now double membrane structures. Now, these membranes are very different than the structures of the endomembrane system. We can't have any vesicles fusing with mitochondria or chloroplasts. So that gives you an idea right there that the structure is definitely different. So this we've talked about a little bit, but then the question is, how did this happen? Okay, so how did the endomembrane system actually evolve? We believe that this happened through a process of invagination as opposed to endosymbiosis. So, in a prokaryotic cell, basically we've got some genetic material and we've got some ribosomes sticking around, hanging out on the membrane. And remember, prokaryotic cells, we have no membrane enclosed compartments at this point. So everything is all in the same compartment. Now, why this happened, we don't really know, but at some point, there was an invagination of this plasma membrane. So if you follow this around, you can see this is all still one membrane, and it has simply invaginated, and it surrounded that genetic material, that DNA, and then brought this portion with it with other invaginations that eventually led to the beginnings of the endomembrane system. So what we have here are membrane-bound compartments that all share the same membrane, which means that we're going to be able to have transport and fusion of vesicles and things of that nature between them. The nucleus did end up with a double layer, but you can see that unlike this one where we have two totally separate membranes, we had the cell membrane here and then wrapped around with this cell membrane. The double layer of the nuclear envelope here is actually just a continuation of this exact same membrane. So a little bit different there. So we know that our ribosomes are in the cytosol. So we don't have ribosomes anywhere else. And we know that ribosomes are required to synthesize proteins, which then makes us say, okay, if all proteins are made in the cytosol, how do they get where they need to go? Because we have some proteins that need to go to the nucleus, and we have some that need to go into a chloroplast or a mitochondria, and we have others that need to move into the endoplasmic reticulum, and then we've got others that need to move through the Golgi and end up at the cell surface. But they're all synthesized in the cytosol. So what exactly is telling them to go to these particular compartments? These proteins are going to be moved to particular portions of the cell due to what are called signal sequences or patches. These are encoded in the amino acid sequence, and they are what are going to direct the protein to where it goes in the cell. Now, if a protein is synthesized and there is not a signal sequence or a signal patch, then it will stay in the cytosol. So cytosolic proteins are lacking a signal that tells them to go somewhere else. That's why they stay in the cytosol. It's essentially a lack of another signal saying, go to the ER or go over here, that means that they will end up staying in the cytosol. Now, these organelles, these membrane-bound organelles, have the same principles as the plasma membrane. So generally, we're not going to see permeability to any type of large or hydrophilic molecule like a protein. So that means all of these proteins are going to need specialized transport. And there are three different mechanisms to move proteins in the cell. The first is called gated transport, and it is going to function to move things in and out of the nucleus. Then we've got what's called transmembrane transport, which is also called translocation because it is required, or it requires protein translocators. This means that we are going to transport proteins across the membrane. And so when we talk about the movement into chloroplasts, mitochondria, peroxisomes, and the ER, That is all going to be translocation. Then we have vesicular transport that can happen within that endomembrane system, which is all going to start in the ER and then move from there. So we have 3 different mechanisms to get these proteins that are all made here in the cytosol into the different components of the cell. Here you can see the different areas that these types of transports occur in and the directionality. So gated transport is a 2-way transport between the nucleus and the cytosol. That's the only place that we're going to see gated transport. Transmembrane or translocation is going to occur in a 1-way direction from the cytosol into peroxisomes, chloroplasts, the endoplasmic reticulum, and mitochondria. Vesicular transport then can go in multiple directions, so it can go in or out, and is all going to originate and start here in the ER. Then we move into the Golgi, we can go into secretory vesicles, we can go into endosomes, lysosomes, we can move things directly to the cell surface, we can bring things in from the cell surface that need to come in. So we have a lot of movement within the vesicular transport. So the signal sequences are encoded in that amino acid sequence. So, going back to the concept of the importance of that primary sequence, that primary structure of the protein, which requires that correct encoding of that amino acid sequence, you can begin to think that if these sequences are encoded in that chain of amino acids, if we have a mistake in there, we could end up with a protein that does not go where it is supposed to go, or maybe ends up in a place that it's not supposed to be. So here's a couple of the different types of sequences. So this one is going to say, take me into the ER. Okay, so when we see this, we've got take me into the ER. And then we've got another one that says, hey, keep me in the ER. Basically, you have to tell the protein everything you want it to do. So just saying go to the ER does not necessarily mean that it will be there forever. And the reason is that oftentimes, once the signal has directed a protein to the given area in the cell, it is actually cleaved off of the protein. So there is a class of enzymes called signal peptidases that are going to cleave off signal sequences. So once you've moved something to the ER, then that gets cut off. And so if we want it to stay there, then it's going to need another signal that says, keep me in the ER. Okay, so we've got the encoding of these in the amino acid sequence. And you can see that a lot of times it's going to be, similarly a chain of different positives, or the green would be a bunch of different hydrophobic ones, things like that. We are also going to see that we're going to then need a class of receptors called signal receptors. So it doesn't help us to have a signal sequence if we can't then have something that recognizes and then will bind to that signal sequence. So how do we find this? We utilized recombinant DNA techniques. So we have a cell here that has a protein in the lumen of the ER. So obviously this protein was synthesized out in the cytosol, imported into the ER, and then retained in the ER due to this signal sequence here. And this cytosolic protein did not have a signal sequence and therefore stayed in the cytosol. So what happens if we take this ER retention signal and move it to this cytosolic protein? The protein moved. What we ended up with is a cytosolic protein sitting in the lumen of the ER, and then an protein that did not have a signal sequence anymore, so ended up staying in the cytosol. So we were able to determine that these signal sequences were what was definitively directing the protein to where it is supposed to go. If you remove a signal sequence, if you modify the signal sequence, if you add in a different signal sequence, that is what will direct the protein to where it is going to go. Now, signal sequences is kind of an umbrella term. However, we do utilize it for anything that is on the N or the C terminus. So a signal sequence, by definition, is a chain of amino acids that is found on the N terminus or the C terminus. Okay, so when the protein folds, that signal sequence is still right there. So nothing has really changed. This type of signal sequence could be utilized in either the folded or the unfolded protein because it does not change. There's another type of signal sequence that's called a signal patch. Here you have different regions of amino acids that contribute to what's called the signal patch. But obviously, when they're spread out like this in the unfolded protein, they're not actually creating the patch, which means that the protein has to fold properly before we actually get the signal sequence. If a protein contains a signal patch, it has to be folded before the signal is actually generated. So anything that requires an unfolded protein would not be able to utilize a signal patch.